Welcome to Medicine and Healthcare Around the World, the podcast where we connect with passionate people from all over the world that are making an impact within the field of medicine and health. So, if you're tuning in to listen to one of our many great stories from wherever you are and wherever you're headed, please be welcome, friend, as we're about to get on with today's episode.
But this opened up a bigger question. This is a humanitarian effort now. So am I going to try to make money from this? What am I going to do?
Dr. Kishor Wasan's work in pharmaceutical sciences has been recognized with over 25 awards and has published more than 550 research papers focusing on how drugs are delivered in the body. As the former Associate Dean of Research at the University of British Columbia in Vancouver, Canada, and the co-founder of the Neglected Global Disease Initiative at the same university, Dr. Kishor Wasan has dedicated a large part of his life to finding solutions for diseases that often get overlooked. In particular, he's developing an oral, less toxic form of a drug called Amphotericin B, which is currently in clinical trials to treat Leishmaniasis, a neglected tropical disease that devastates many impoverished areas of the world. Now let's jump into the episode and hear the story by Dr. Wasan himself. Well, that sounds great. So yeah, we've started recorded, and Dr. Kishor Wasan, or Dr. Kish, I think, thank you so much for attending this podcast. It's a great honor, and we'll get into the job that you do and the amazing job that you do concerning Leishmaniasis and the drug you've developed. But would you mind just intro-wise tell a little bit about yourself?
Yeah, sure, Hassam. So thank you so much for having me on your wonderful podcast, and good luck. And hopefully you'll be able to spread the word of all the great podcasts that you're doing. So I'm Kishor Wasan. I'm originally from Kingston, Ontario, Canada. My parents were born and raised in India, and then came to Canada in the late 1950s, actually. So I was brought up in Canada, and did my elementary school and my high school and my early years at university in Canada. And I was at Queen's University in Kingston, Ontario. Kingston's a small town just northeast of Toronto, and actually not that far from northern New York and the U.S. border. We used to go over there for American fireworks for July 4th. They had better fireworks than we did back in the day. Um, and so, long story short, I ended up because I had a personal love for the state of Texas and had family down there. I ended up doing a pharmacy degree at the University of Texas at Austin and worked as a decentralized clinical pharmacist in Dallas for a number of years, where I was particularly involved with infectious disease patients, cancer patients, patients with HIV/AIDS, patients in that area. And I worked side by side up on the floor in the unit with physicians and nurses that were dealing with some really tough cases of SAM, tough patients, multi-factor conditions. I was in charge of making up the medications and helping get those medications into patients. I was also part of the advanced cardio life support team. So when there was a code, a patient went to cardiac arrest. I was part of that team, which was pretty traumatic, but also exhilarating in trying to help patients. And that got me interested in saying I want to go on and study the science of really cancer research, and so on. And with a couple of physicians' help at the hospital in Dallas, ended up in graduate school at the MD Anderson Cancer Center at the University of Texas, and of my supervisors, Dr. Gabriel Lopez, and Gabe and I have stayed in touch actually the our whole time that I did my degree and moved on. After doing a doctoral degree under Dr. Lopez's tutelage, and I actually did my PhD on liposomal amphotericin B, and we can get into amphotericin B a little bit better because this seems to be a drug that I can't get away from. I mean, I wanted to do other things in my life, but amphotericin B became sort of my thing. My kids make the joke that when I pass away on my tombstone, they'll call me Amphotericin B man because it seems to be the drug that I've spent my whole entire career on. Um, After finishing a PhD and doctoral work and clinical doctoral work with Dr. Lopez, I did a postdoctoral fellowship at the Cleveland Clinic with Dr. Richard Morton, and the Cleveland Clinic is world renowned as well, particularly in lipid and lipoprotein metabolism. And then I was recruited back to Canada to the University of British Columbia and worked through the ranks at UBC. Went from an assistant professor, had a nice lab, got peer review funding, built up my whole lab, and was the department head for pharmaceutics, and then was became associate dean of research. And then I was recruited to be the dean of the pharmacy school on the prairies, and did that for a stint. And then I was going to retire, but then I already still had an appointment at UBC, so I got called back to UBC, and that's where I've been since about during the pandemic around 2020 when I retired after my deanship. So I'm sort of out of retirement, I guess. And I've been working at the Vancouver Prostate Center in Urological Sciences, so I do prostate cancer research. But I also still am involved in infectious disease and leishmaniasis, neglected tropical diseases. During my time at UBC, a colleague of mine, Dr. Bob Hancock, and Bob is a world leader in microbiology, formed the Neglected Global Diseases Initiative. We started that around 2009, 2010. And when I went to become a dean for a few years, my colleague, Dr. Richard Lester, who's an amazing infectious disease doctor and is doing amazing work in Rwanda, took over the reins, and I stayed on the board. But I'm back now as still the co-director of NGDI, and in fact, we're about to have my term is about to end, and we're about to have a new younger generation take over in 2026. We haven't done a formal announcement yet, so I can't name that. So that's my background. I'm a pharmaceutical scientist, a drug delivery, clinical pharmacologist background with a clinical pharmacy background as well. So I bridge between basic science and clinical science.
That's right. And here we have the kind of neglected tropical diseases, and we'll talk about how your drug fits into the whole puzzle, because I'm really amazed by the kind of the important decision that you had to make at a certain point in your life. But the Global Health Initiative of British Columbia is like an institute, from what I understand, and I
It's
think
an
just
initiative. We're not really an
oh
institute per se. We're more just a loose network of folks with a common interest around neglected global diseases,
oh
including
I stand
students.
corrected.
Which, yeah. Oh no, no,
Initiative.
that's okay. A
Yeah,
lot of
yeah.
people
Uh,
get that mixed up.
yeah, so the initiative in this case, and I think I haven't come across this in any other part in kind of tropical global in the neglected tropical diseases, but you are coining the term kind of tropical global diseases. Sorry, forgive me, that was wrong actually. Neglected global diseases instead of neglected tropical diseases. And I'd like to ask you, before we go into the story of amphotericin B and all of that, why you choose to coin it that way, the term.
Yeah, so that's an excellent question, Hassan. And there was a lot of discussion and debate at UBC with our colleagues about, and we strategically called it neglected global diseases on purpose, as opposed to neglected tropical diseases. So there is a reason and strategy behind it. And it turns out now in 2025 that what we were thinking about in 2009, 2010 may have turned out to be pretty smart on our behalf, or we were just very, very lucky. Um, in the following sense. So, as you know, the neglected tropical diseases, as defined by the WHO, or the World Health Organization, are 21 infectious diseases that occur in majorly the tropics, things like Leishmaniasis, dengue fever, African sleeping sickness, and I can go on. There's a whole Noma has just become a new one, the 21st. And the WHO is this wonderful description of neglected tropical diseases. And these diseases happen primarily in the tropics, and are usually, the definition is the proportionality between the magnitude of the disease, the number of people that are actually infected by the disease, versus the resources that are been given to try to treat the disease, is completely out of proportion, Hassan, completely out of whack. I mean, we don't have that's why they're neglected. And so that's why they're neglected tropical diseases. But UBC wanted to be a little bit different on two fronts. Number one, we recognize that, what is the main driving force behind neglected? Neglected is not just that there's this disproportionality between resources and the magnitude of the disease, but it's neglected people. So, for instance, we have people that have malaria or drug resistant tuberculosis or HIV AIDS that may not be considered a neglected disease, because there's been a lot of neglected tropical disease because it doesn't occur just in the tropics, but also because there has been a lot of money put toward them, but they're still neglected because neglected people. So, even in our own backyard here in Canada, unfortunately, our Indigenous population um, has a very high rate of drug resistant tuberculosis, which actually matches what you might see in a low and middle income country. It's a terrible situation, even here in Canada, which we need to do a much better job on. That's because they're neglected people. So the strategy was, the reason why we called it neglected global diseases is because what we're focusing on is the neglect of neglected people having these diseases. And so, therefore, because we're also doing some great research at UBC in the areas of malaria and drug resistant tuberculosis, we knew that neglected global diseases was a very important term. And that's why we've termed it that way. The other thing, and this is, as I made the comment, 2025, it's become very important now in 2025. In 2009, we may have been blue skying or crystal balling this, but we felt that these diseases were going to be moving out of the tropics at some point. And as it turned out, that turns out to, unfortunately, to be true because of climate change. So different parts of the world that never saw these diseases are now showing up in those parts of the world. So I'll give you a specific example. So cutaneous leishmaniasis, this is the skin lesion on your face from the scanfly, has now been identified as an epidemic in the southern United States, of all places. In fact, at the American Society of Tropical Medicine meeting a few years ago, they actually made a big announcement around that. And there have been a variety of papers talking about how CL, cutaneous leishmaniasis, might actually be an epidemic in the southern United States. We're talking places like Louisiana and Texas and Florida and Mississippi. There are cases of leishmaniasis showing up down in those parts of the United States, never before. And the reason why is the environment and the climate is now made it fertile for the sandfly. This is the parasite that causes you to get leash. Is now ripe for growth, for reproduction, for spreading, for so on and so forth. And now we're getting CL in the United States, um, where you wouldn't consider that a tropic area. So it's a neglected disease that's showing up in the United States. So that's why we call it a neglected global disease. And I think the last comment I would make is, because our world is so interconnected now, Hassan, the fact that it's easier to travel around the world. And this has become an issue now here in Canada, even a cold climate here, like in Canada. Um, we have people from different parts of the world that are unfortunately bringing these diseases when they fly over here, and they're showing up in cases. My mother is a retired forensic pathologist, and she did her degree in India, where our family is from originally. She actually, when it saw cases when she was a working physician, and this is back in the 80s and and in the 90s of people that were having malaria and other types of infectious disease that you would never have seen in Canada. And in fact, the only my mom is, I mean, shout out to my mother. My mom was one of the top forensic pathologists in Canada back in her day, but she was also very smart because she could identify these diseases that other trained Canadian physicians couldn't because they never saw it before. So that's a long answer to your question, but there is a strategy and a rationale behind. We call it neglected global diseases as opposed to neglected tropical diseases. We want to be more encompassing. We want to be able to
Right.
include other diseases, and we want to focus on the people who are neglected.
That's right. And I would say reasonably so, because like you said, in Louisiana, I think you mentioned that state in the US where you've seen the increase of subcutaneous leishmaniasis. And just to give the people listening to the podcast, I'll probably put a few pictures up, but you have the kind of subcutaneous version of leishmaniasis. And then there's the, which, like, yeah, you get really
The
ugly
systemic
looking
blood
ulcers.
version, yeah, called visceral
Exactly.
leishmaniasis. And in
Uh,
fact, Hassan, you'll love this.
yeah.
After Hurricane Katrina in 2008 in Louisiana, I was invited down to give a talk in 2009, and in the ninth ward in Louisiana, they were still
Right.
cleaning up in New Orleans. They were still cleaning up from the hurricane. They had cases of VL in
Oh,
Louisiana. Not
there
just
you
CL,
go.
not just cutaneous, but the blood-borne of leishmaniasis in
Yeah. So
2009.
yeah, just to touch on that too, you have the kind of subcutaneous, but then the visceral version, which is the more deadlier one that you really have to watch out for. But I would like to ask you, and please correct me if I'm wrong on this, if there is the kind of spreading, because one of the part of why it's so neglected is because it's also about the kind of socioeconomic situation that we see in a lot of these developing countries. And there's the pharma, of course, that has to make a profit. And of course, they focus on the recipients that are able to pay for the medication. And would you agree that part of it, why it's so neglected, is also, it's now you said it's global, and I would agree with that. Previously, maybe people would think in the tropical regions like sub-Saharan Africa, but would you also agree that it is neglected because there's not much economic focus, let's say, on developing the necessary drugs to treat these illnesses?
Well, I think infectious disease, regardless of if it's neglected or not, and COVID-19, I think, has brought this to light, has been underdeveloped with new therapeutic drugs, new therapeutic indications, new therapeutic targets for almost 20 years. And I've been I mean, and now this is a little self-serving, but I've been in the infectious disease area for over 35 years. And we've been clamoring to, and there's been some really great research at the biotech level and at the university academic level, grassroots level, some amazing research going on. Um, but it has not bubbled up to the pharmaceutical industry wanting to take these projects on and develop it as medications. And exactly to your point, Hassan, because the cost benefit analysis from a farmer's perspective um, is not there. And what I mean by the cost benefit analysis, the amount of money it takes to make and develop a new drug. And I'm a pharmaceutical scientist. And the majority of my students are in big pharma. So big pharma has done some amazing things, and it's not the evil. It's not a black white situation. It's not the evil empire everybody thinks. But at the same time, they have not, and that we're quite not happy about this because from a cost benefit analysis, benefit being their economic benefit and the cost of making it, they're not seeing that it's worth putting the amount of money and resources for the benefits that they would have. So big pharma has not been, developing and investigating new therapeutics for a variety of infectious diseases, even the ones that are not what we call neglected, and that is a big problem. However, Hassan, things have gotten. I'm a man of hope and faith. So, um, and in these trying times, we need hope and faith. But what I would say to you is, big pharma is a lot better today with helping out with neglected diseases and with infectious diseases than they were when we started NGDI back in 2009. A number of them have started up global health programs within their companies. A number of them have partnered with the Gates Foundation, the Rockefeller Foundation, and other organizations around the globe to provide either medications that have come off patent at below costs or have actually provided resources and money through group grants to help support organizations that were not around 20 years ago, that are now around, that do humanitarian work around the development of new therapeutics or modified therapeutics for neglected diseases and infectious diseases. So big pharma is starting to play a bigger role through that back door. But you're correct, from the front door's perspective, not many of them have decided to jump into infectious disease because it's not cost effective. Now, I think that might change because of I think COVID-19, if nothing else, has awoken the world to realize that we were not as well prepared for a pandemic as we thought we were. Our infrastructures and our supply chain management was actually quite fragile, and that became exposed during COVID-19. And so, moving beyond what happened in that time, I think that there has been a people are rebooting on how they want to approach things, and I do see better things now with pharma than I did back in 2009, 2008, 2009. So there has been improvement, but you're right. They're not full force yet to the level that they could probably be.
But
And if they took
maybe,
1% of their
yeah,
budget and put it dedicated to NGD, the neglected global disease work, we could make a huge impact on the world.
that will be an amazing impact for sure. But I would maybe argue that you're in the forefront of changing this landscape a little bit, and I'd like to go into the drug that you and your team has developed, the kind of
Well, it's a
amphotericin.
modified drug delivery system for a very old drug, Amphotericin B, that was actually discovered in the 1950s. But yeah, so we've got a novel technology to improve its ability to do its thing. So just so we can clarify that,
For
which
sure, for
actually is
sure.
important because it makes drug development faster in a lot of respects and a little bit
Yeah.
easier than a brand new molecule from scratch.
I can imagine that it absolutely streamlines the whole process. But your team, I would say, is, and this is why so I'm amazed by it, the kind of changing the landscape uh, of the treating people with neglected tropical diseases. And in the case of amphotericin B, it is used as a treatment for Leishmaniasis. But that was not, from what I understand, originally the thought when you and your team were behind the
Well,
kind
let me
of.
tell you, yeah, if you can indulge me, let me tell you the story about how this all started because
Please.
there is a story about the development of the oral amphotericin B and where we're at, but there's also another underlying story for your audience that's actually very important about my evolvement, my evolution, and about drug drug development, particularly for neglected diseases, and my thinking about how everything has happened. And I had to change my ways personally. It was I don't know if it was an epiphany, but I came to realize that what was I doing with my life, quite frankly. And finally, my mom is now happy with me because, so there's actually a two pronged story here. So if with your approval, I'll tell you the story and how they
Please go ahead.
interweave. So I am a classic pharmaceutical scientist academic by training. And when I got to UBC and started my lab, and this is not an indictment on academia, this is just the way academia works, is as a young assistant professor without tenure, the goal is publish or perish, train graduate students, be a good citizen and service, also do a lot of teaching, and get money, bring in money to do grants, to train students, to publish publications, and so on and so forth. And because I was in the pharmaceutical sciences faculty, the majority of my students were set up to go to pharmaceutical industry. Some went into academia, some went into other areas, but the vast majority of my students ended up with jobs in the pharmaceutical industry, either in a small biotech company, or a lot of them in big pharma. And so I was your classic pharmaceutical scientist. Let's publish our work. Let's promote it at the pharmaceutical sciences meetings. Let's see if we can get a big pharmaceutical company involved. And let's see if maybe we should start our own company. Let's get patents. Let's make money, and so on and so forth. So that was the original dream, and that was what I really was trained to do. And for over 20 years, that's what I did. I mean, and I was very fortunate that I've had an amazing career, which you know, far beyond my dreams, that I will, um, far beyond, sorry about that, far
Oh,
beyond my
don't worry about
dreams,
it.
that I never envisioned, and had a wonderful, wonderful career. But where this all verged and changed was started in around, believe it or not, 1999. So almost over 25 years ago. And so, as I mentioned to you, I did my doctoral work at MD Anderson Cancer Center with Dr. Gabriel Lopez Berenstein. And Gabe was involved in the development of a lipid-based formulation for amphotericin B. Amphotericin B is an antifungal, antiparasitic drug developed for the treatment of bloodborne fungal infections and parasitic infections like Leishmaniasis. And it was given intravenously, but its major concern was what we call dose dependent kidney toxicity. And so, if you gave infusions, as you increase the dose, patients would get kidney toxic. And if you were dealing with patients that had infections, fungal infections, secondary to being immune compromised or having HIV/AIDS or uncontrolled diabetics and so on and so forth, then having kidney damage as a result of the drug was not a good thing. It just made things more terrible. So Dr. Lopez developed, and I was his graduate student at the time, developed, and we published a number of papers together, developed amphotericin B lipid complex, which is now became Abelset, which came on the market in the mid 1990s. Subsequently, Ambisome was developed, which is a true liposomal drug. Abelset's actually a lipid complex, and it's actually now gotten the majority of the market for as the amphotericin B product given parenterally. So the reason why I tell you this is because in 1999, 2000, 1999, I was giving grand rounds at Vancouver General Hospital to the infectious disease group. And at that time, there was a lot of interest in combination therapy of amphotericin B with drugs like caspofungin and other ones like that to treat difficult fungal infections in a hospital setting. But also, there was a lot of concern, particularly from our transplantation physicians, that patients were getting systemic fungal infections prior to transplant. So they were being forced, as you're a medical student yourself, you would understand, they were actually making patients immune compromised on purpose so they wouldn't get organ rejection when they were doing, a lung kidney transplant or whatever. But
That's right.
then they were open to fungal infections. So what would happen is these patients would have to come into the hospital a few days before the transplant, and would have to get IV infusion of amphotericin B prophylactically at lower doses so that they wouldn't develop these fungal infections before the transplant. But as you know, if you bring them into a hospital a few days before the transplant, that's more it's not cost effective, it's more expensive. They could get a nosocomial or hospital-borne infection because they're in the hospital. They're already immune compromised, and then they have to be on this stupid IV infusion of liposomal amphotericin B, which is a great drug, but it also has side effects because if you don't infuse the drug properly, you can get chills and shivers, and you can get toxicity secondary to the administration of the drug itself, red blood cell hemolysis, whole pile of things. So it was a real problem. So I was giving this talk at the back in the back of the room. Some physician said to me, said, "Could you develop an oral formulation of amphotericin B prophylactically that we could give to transplantation patients who were going to be candidates for transplant prior to them coming to the hospital. So the idea was that they would be able to take this oral, this cost-effective, safe, accessible uh, oral AMP at home, maybe a week in advance, kind of like you give an antibiotic to a kid when you're treating an infection. But this would be on board in advance as a prophylactic to build up the levels of amphotericin B in the tissues where the fungal infection could occur. And AMPI has some very unusual pharmacokinetics, and we can get into that because that people don't understand that. It's about systemic exposure, not necessarily blood levels. But could you do that? So as I was driving back from rounds, back from the hospital to my lab on campus, I was thinking to myself, how could I do this? And to be quite honest with you, Sam, I didn't think it was possible. And that was because the molecular weight of amphotericin B is too large. It gets broken down and chemically broken down in the stomach because the stomach has a acidic pH, so it would break it down. I didn't think you could get enough absorbed at a high enough level for a sustained period of time for it to have pharmacologic effect. And I was always worried about not only the kidney toxicity, but potential liver toxicity, because if you're getting it absorbed from the upper GI tract, it goes
Has
through
to
the
pass
portal
through the
vein and
liver.
into the liver. And then you could even have GI toxicity. So you were not only introducing potential toxicity from the kidney itself because of the nature of AMP, but now you, because of the route of administration, you might be introducing new toxicities, let alone were you going to be able to get enough absorbed and enough bioavailable in the systemic circulation, and more importantly, in the tissues where the disease occurs to have pharmacologic effects. So I didn't think it was possible. But at the time in our lab, we were working on using lipids, basically dietary lipids, mono and diglycerides, triglycerides, to improve the absorption of lipophilic drugs and through a mechanism called lymphatic transport of drugs. This is something we were doing. I had a small grant. There were some leaders in the field that were already doing that work for lots of different drugs. So I thought, maybe we can do something. And of course, when I walked back into the lab, I had this weird look on my face, and my students went, "Oh no, Keisha's got some another crazy idea." Because from every time I came back from one of these talks, right, or a conference or something, I'm always like, "Oh, we gotta try this," and but that's science, right? That's what makes science exciting. But this was very practical, right? Could we have developed a prophylactic oral formulation of amphotericin B that was cost effective and safe, and was accessible, that could build up a depot effect, and had enough systemic exposure at non-toxic doses to prevent fungal infections from growing in these patients. All right, before they got the transplant. So that was what the mission was. So, to make a long story short, we had a number of fungal models already established in my lab. As I told you, I was an amphotericin B guy because I did all this work at MD Anderson Cancer Center, and I was drawn back into AMP again. And I thought, oh God, I can't believe I'm doing amphotericin B. But okay, so drawn back into AMP again. I knew how to establish the fungal models in my lab because I had done this work at MD Anderson Cancer Center. So we had the fungal models. Long story short, we developed an oral formulation. It seemed to work really well in Candida albicans and aspergillus. And we published our papers, and we filed our patent with our provisional patent with the tech transfer office at UBC. And we were, I was about thinking about starting a company. And then back in those days, um, the joke was on campus, what UBC professor doesn't have their own spin-off company from their technology? Because everybody was doing it because it was another way of getting money for the lab, but it was a way to translate the work to the real world. And that's a good thing. I mean, yeah, universities, there's a lot of criticism these days. As you know, academia is under a lot of scrutiny. What is the worth of academia to the world? Um, the one nice thing is, in my world, in my little sub world of our people, we were all doing work that was practical to translate new standard of care to people on the ground to solve a practical problem. So I was trying to solve a practical problem that this infectious disease doc wanted, which was, could he develop a prophylactic oral amphotericin B to help treat patients in advance so they didn't get fungal infections when they were going to get their transplant. So it was a very practical, solving a practical problem.
Just a quick break and we'll head right back into the episode.
So we filed our patents. We're going to start a company. Things are looking great. I was going to do the classic, route that everybody does. And then one day, my whole life changed. It all really changed, Hassan. So a medical student, just like yourself, came to my office, said, "Dr. Wasan, I saw your press. We got a little bit of press and your papers and some your announcement about your oral amp B work. Have you thought about this devastating disease called Leishmaniasis?" And I looked at him and said, "Leishman what?" And I was embarrassed because I didn't know what Leishmaniasis was. And what made this even more embarrassing is at that time, it's better now, Leishmaniasis was an epidemic in my mother country of India. So I could hear my mother's voice. What are you doing? You develop this development and you're not even helping people in your own country? Like, who are you? I could hear my mom getting mad at me. But I was super embarrassed. So when the student left my office, I madly looked up leishmaniasis. I said, "What is this disease? What is going on?" And what we turned out was that there's this thing called leishmaniasis. And visceral, as you mentioned at the beginning of the podcast, is the bloodborne parasite that the sandfly stings you. And the sandfly grows in regions of the world where there's poor sanitation, very tropic, a lot of it in rural communities. So at that time, it was places like Calcutta and the Bihar region of India. It was definitely in South Sudan. It was in Ethiopia. We had it in Brazil. I can go on and on and on. And this disease, there was not really good treatments. There was liposomal AMP, but it wasn't cost effective. It was hard. It was not accessible. It wasn't tropically stable. People didn't have an ability because many of the people who were affected with leishmaniasis were women of childbearing age and children, and they could not leave the rural communities to go to a medical clinic. Plus it was expensive, and you have to sit there with an IV infusion. The other oral drugs that were available all had major toxicity and stability issues. There was also fake drugs out there. There was piracy going on. It was crazy. So what this medical student was saying to me, if you could develop an oral formulation of amphotericin B, because we knew amphotericin B, the molecule, the drug, was effective in treating, visceral leishmaniasis, this blood-borne parasite from the sandfly that stung you, and the skin infection, cutaneous leishmaniasis, which even though it isn't life-threatening, has a big social barrier for people that have the disease. It can lead to something even more serious. So don't underestimate CL. It's a very, very serious disease, and even to this day, we don't really have any good treatments. So could we develop something that would develop an oral amphotericin B? So I said, "Oh my God, maybe we should start looking into this." So I madly did research on the area. I tried to find funding for it. I was invited down to Seattle to the Bill and Melinda Gates Foundation to give a talk about what we had done, and they partnered us with one of their partners that they had been funding, the Consortium for Parasitic Drug Development out of North Carolina that was doing work in dengue fever, African sleeping sickness, leishmaniasis, and they had established animal models of VL. And so that's where we started to use our oral AMP. But this opened up a bigger question. This is a humanitarian effort now. So am I going to try to make money from this? What am I going to do? Turned out that UBC at the exact same time was developing global access and equity principles, that they were setting up principles saying that if you wanted to develop a new standard of care, a technology, a diagnostic, a drug, a vaccine, at or below cost for low and middle income countries, we would support that at UBC. That would be part of our global access and equity principles. And so when, already my patent had been filed for oral Amp B for fungal infections, we the tech transfer office said, "Quiche, have you thought about maybe oral Amp B for Leishmaniasis under global access and equity?" And I thought to myself, and this is where my life changed. I said to myself, all right, but then, I was a full professor and I was getting all my fancy little awards and I was getting notoriety for the work we did, the classic academic. But was I really making a difference in people's lives? Was I really doing what I was supposed to be doing at a university? And so I had an epiphany. I had two choices at that point. I could have just started my own biotech company like everybody else and then, got the investors and did all that kind of stuff, have my own company and and you know try to be rich and do all that kind of thing and so on, and then hopefully have a big pharma buy out my biotech company with a big payment and do all those things, and that's what a lot of people were doing, and that's not a bad thing. We've got a lot of great technologies out of UBC that have become actual drugs on the market through that mechanism, so it's not all bad. Or I could do this for humanitarian reasons, and I actually felt terrible. I said, "What have I been doing? Have I really made a difference in people's lives? If I really am I really doing what I'm supposed to be doing?" And that's when I said, "You know what? I'm not going to start my own biotech company. I'm not going to lead something like that. I want to do this for humanity. I want to do something that is going to make an impact." So I said to the tech transfer office, and we got lucky because with the oral amphotericin B, you can almost have your cake and eat it too. For the fungal infections like vaginal candidiasis in women that affect women all over the world, and some of these other fungal infections, you could make money. You could make it as a normal product and do that. But for leishmaniasis, I said, let's develop it at or below cost for low and middle income countries and make it humanitarian. All we want's cost recovery on its development. I don't want to have any profit, nothing. And so we signed up for the global access principles. And with the Gates Foundation, we got preliminary data and got some really exciting results. And one thing important in science is when a third party validates your work and their models, that shows your science is good. Okay. I mean, these days there's so much fake science out there and the science is under attack and people having a hard time reproducing the work. And there's all sorts of things going on. Some of it is correct and some of it is false accusations. So it's all over the map. The best way to show that your science is strong is to have an independent lab validate your work. So what we did is we took our formulation that we had shown in our animal models and fungal models and provided it to the Gates people. My postdoc went to Columbus, Ohio, Ohio State University. Dr. Carl Werbowitz, who was funded through the Gates Foundation through the consortium, had a validated VL model. By the way, this is all published, so you can look it up on PubMed. In his lab, I sent my postdoc with the formulation there, and they administered it, double blinded. And the results in their VL model was even better than my results in the fungal model. We got 99% reduction in the parasitic load in 5 days. It was as good as the liposomal formulation at non-toxic doses. So everyone became really, really excited about, okay, we might have something with oral Amp B for VL. And and then we had to develop a tropically stable form because the original formulation was not supposed to be tropically stable. Okay. So we reformulated it. My wife was involved and she's a co-inventor and she's a brilliant drug formulation scientist. So it's a family affair here. My wife, Dr. Ellen Wasan, developed the 2.0 formulation, and it worked as well as the original one, and we published that. And through the consortium parasitic drug development, we got all the preclinical done. But then the next stage was, how do you get it to development? How do you get it to clinical development? And luckily, we had a local biotech company, Ico Therapeutics, that was willing to sign on to the global access principles at UBC, which said that for the Leishmaniasis indications, they would help develop it at or below cost. For the fungal infection indications, that's where they said they would like to be able to commercialize it. So we were able to do both. So we gave it to ICO. ICO was able to get it through GLP toxicology in dogs and was able to finish phase 1 human safety clinical studies, both single dose and multiple dose in patients. But then two things happened. Number one, COVID hit. So we were all shut down and nobody could do a phase 2 trial. And number two, unfortunately, ICO ran out of cash and, they're a small little biotech company. And, they did a great job and got us all the way to the end of phase 1, but we're no more. So just recently, and I think I sent you the link through LinkedIn, so you're welcome to share that. The
I'll make sure
license
to do it.
came back to UBC, and now we have licensed it to a new local biotech a new company, Luxidex. Dr. James Kim, who's a chemist by training, so he's a scientist himself, but he's a CEO who is now willing to take it on through the clinical development, but has also agreed to make sure that we still do the global access equity principles at or below cost for Leishmaniasis and for developing world indications. And I think he's even willing to do it for some of the fungal infections at or below cost. You could read the press release. It literally came out yesterday. And so we're very excited about that. James also has a breadth of colleagues in Korea, in other parts of the world that might be interested to get us the dollars. Because what we need to do, Hassan, is we need to find the resources and dollars now in a very tough time, as you know, what's been happening in the world with global health funding and so on. And I don't like to be political, so I'm not going to stay out of the politics of it. But I think we all know what's happening right now with funding for global health programs around the world.
Yeah.
In a difficult time, we have to try to find unusual ways of get things funded. So this announcement with James couldn't come at a better time, and he has new resources and new opportunities for us to get this into phase 2. So we need to get this oral AMPI into phase 2, phase 3 clinical trials in patients that actually have VL in countries that are showing still a big problem with VL. And we're looking at Ethiopia and we're looking at a few other places. What stuns me more than anything else is that this has taken so long to get to this stage. We had thought by now that there would be other good products out there to deal with VL and with CL and with other fungal infections. Goes back to our original discussion. You were asking the very good question, why pharma doesn't seem to be developing new drugs or doing new research in infectious disease. Leishmaniasis is one of them. There's been some new molecules. A couple of organizations have gotten something in phase 2, but we're still using all the old drugs: miltefosine, paromomycin, antimonal therapies, liposomal AMP, all the same drugs that we were looking at for VL back when Andrew Gray, the medical student, walked into my office that so many years ago and said, "Have you thought about an oral AMP for VL?" are still being used today. There hasn't been that much of an advancement. And so an oral AMP product, according to a lot of people, we got support from Doctors Without Borders Canada, the WHO NTD working group. Their working group has endorsed a development of an oral AMP for pediatrics in VL. They wrote a big report on it. That's out there in the web. Still think to this day that an oral AMP would be very important in different aspects of Leishmaniasis. So that's why we want to develop it. If there was something out there that was already better or that we didn't need an oral AMP, I would have said, let's move on to other things. Because I do other research. My wife does vaccine research. I do prostate cancer research. And oral AMP, we could have done it for the fungal infection indications where there's still a need, like vaginal candidiasis in women. We wouldn't have done the VL. We would have said, okay, there is, or CL. And CL is even worse. Apparently, there's not very many good treatments for CL. The reason why we're still in the game with Amphotericin B is not because it's just my true love of this molecule that I've done, that I've worked on for all my life now, it's because there's still the need. So this is a
Yeah.
two-pronged story, Hassan. There's the story of the actual development of oral AMP and its usages, but the real underlying story to your audience, to students, to other people out there to inspire them is what we're doing with the technology. As I said, I could have formed a company. I could have done the classic route, biotech company and do it that way. I did it. All right. We are doing it for humanitarian reasons. And I don't have any stock in James's company. You know, and even James, any company that's involved with oral AMP has to sign or agree to the global health access equity and principles that UBC has laid out, or
Mm.
UBC wouldn't license the technology to the company. And if you read in James's press release yesterday, his press release, he makes a big statement right at the beginning of the press release that, I'm going to follow the global access, health, and equity principles of UBC as we try to develop it. Okay. So I didn't start my own company. We've only given it to company. You need companies to help you clinical development. You have to, because they can bring in the big dollars, right?
Sure.
Neglected global diseases can't do that. So that's the story of that's the story of AMP. And then that story led to NGDI. So as we were looking at developing oral AMP and going through all the things I had to do to get grants and money and so on and so forth for this neglected disease, I thought to myself, I wonder if there's other people on campus that are going through the same craziness we're going through to try to get this developed. So Dr. Bob Hancock and I, we went to our president, Dr. Stephen Toop, our vice president of research at the time, Dr. John Hepburn, my dean, Dr. Bob Sindelar. Were all extremely supportive. And the vice president said, Here, I'll give you a little bit of money, hire a couple of summer students, do an environmental scan on campus to see what's happening. And you won't believe, Hassan, what was happening. What the joke on campus that I was making is there were neglected people on campus, neglected faculty, neglected students, neglected
Right.
staff doing neglected disease research and had nowhere to go. They didn't know how to get funding for their work, how to promote their work, how to connect them with the right partners in low and middle income countries so that they could get their work from the ivory tower of the university out into
Right.
the real world to make a difference. And that's why we started NGDI. And we just celebrated our 15-year anniversary of NGDI. But that's
Congratulations.
why we started it. It was built off the frustration that we went through with oral AMP. Then I found out I was not the only one having that frustration. I was just loud and noisy about it. Everybody else had that same frustration. And we got some amazing work going on in NGDI, drug resistant tuberculosis, sepsis research, telemedicine work in Rwanda, trachoma. It goes and goes on, malaria work. And that's part of the reason why we called it NGDI to your question, than NTD, because a lot of the people that we had on campus, they were neglected people, but they were doing work in drug resistant tuberculosis, in anti-malaria work, in antimicrobial resistance. And those are not under the classic definition of a neglected disease, but they were neglected because these people were neglected and they were working in populations that were neglected. Thus, the Neglected Global Disease Initiative. So that's the story
Love
of
it.
Amphotericin B and the story of how we started NGDI.
And so inspiring.
I
I
don't know.
love
I too
that.
much
Just
talking, but I
no, no,
hopefully
no. Please, please.
questions are what we were
I
trying to
think
do.
the word needs to get out there. Such an inspiring and amazing story. The choice
And
that you
now my
made there.
mom feels that I've actually done something with my life, and I too feel that I finally did what I really did. And I got to tell you, I made it. I made a change in my life. Now it's easier for me now at this stage. One of the questions that comes up to me all the time is, would you have done NGDI? Would you have done NGDI and this humanitarian work if you were not a tenured professor? You were a young assistant professor when this all started. And the answer is, that's very tough because, you have to publish and perish. You have to bring in money. You have to develop technology. Those are the check marks to get promotion and tenure. So it would have been extremely brave of me to try to do it as an assistant professor. It isn't as brave for me to do it once I'm a tenured associate or full professor, which is when most of this thing started to take off. However, what we wanted to do is create an environment so the next generation of students and young assistant professors could go into this work and be credited for it. And I think we have moved that bar. And now there are opportunities out there and ways to highlight your scholarly activity that's different than the traditional ways that now promotion and tenure committees are looking at and counting it. That was not true when I was a young assistant professor back in the 90s and in the early 90s. They wouldn't have counted any of this stuff back in those days, but that's changed. So my answer to your question is that I wasn't that brave because I did this after I was tenured. However, we could move the bar. You know, it's all about evolution, right? There's evolution and there's revolution. You revolt against something short term, things can change, but then it always falls back. If you evolve and develop evolution, it sticks. Things stick when you evolve. And we've evolved. So pharmaceutical companies are now got global health programs. They've evolved better than in the 1990s. Okay. Tenure promotion committees have evolved. There are more global health programs at universities. They've evolved. Okay. There are now non-traditional funding mechanisms to fund this kind of work now than there weren't before. Before there were no places. Gates Foundation was barely going. There was no Clinton Foundation. There was no DNDI. There was no EU Horizon. There was no GHIT. There was no Wright Foundation. There was no mechanisms to fund neglected global disease and global health research. There was, NIH and Canadian Institutes of Health Research in Canada and some of the granting agencies at government levels. All right. And maybe, celebrities and philanthropic people.
Yeah, those two,
I
of
mean,
course.
celebrities who wanted to get their name on something to make them feel good or feel, do a good thing, which is, that's okay. It's good. I don't mind. But there were no mechanisms. But now it's evolved. There are places we can apply. In fact, I'm working on 2 grants as we speak. One is due next week and one's due at the end of March to programs that did not exist. All right. Or money was not put in those programs back in 2000. So we've evolved. Things have gotten better in terms of stuff, but we have a tremendous long way to go. And what I'm hoping to do here is to inspire that next generation. So an assistant professor can go into this area. Graduate students and postdocs can make this their career. I didn't start out as a graduate student and a postdoc with this being my career. Not at all. I mean, to the opposite. If you told me in 1999, 2000, as I was going up for tenure at UBC, that this is where I am in 2025, I would have thought you were smoking something or you were, you're crazy. So,
Right.
that's how
It's
life is. But
it's
for me now, it's a way that I can tie up my legacy and what I'm trying to do. But it's not about me. It's about just
but
inspiring the next generation. It's your
yeah, as
generation, Hassan, that have to do it. My son is probably similar to your age. He's an environmental scientist. My daughter is second year in public policy in back east in Ontario on
right,
university.
right.
It's that generation. It's
And
your generation that have
it's
to take the baton from us now. And we could argue if our generation did a good job or not. And some people don't think we have, some people think we have, but in this little slice of the world, if we could set the scene to help you out, to inspire you, and you can make a difference because you're a physician. You could be a clinician scientist. You could be on the ground making a difference in these countries because you could give the medical care. You could take my oral amp E and give it to patients. I would love to see that happen someday.
hope hopefully it
But
will be orally,
yeah.
from what
And
it
by the way, the
sounds like,
coolest
and
thing
I
that happened
understand.
about a year and a half ago, my son is an agrologist. He's an environmental scientist, a plant soil scientist, and I'm an infectious disease pharmacologist. Totally opposite. My son and I actually published a paper together and actually did a couple of webinars where we looked at the role of soil science in the progression of neglected tropical diseases due to climate change. So I took his work, what he was doing in environmental science, with what I
And
was
you
doing
had
in
not.
NTDs, and we put something together. So it was cool. I published a lot of stuff with my wife, but because we're in similar fields. but I've
Sure.
never published anything with my son. So that was great.
Well, congratulations to that, Dr.
Yeah, oh,
Kish.
well,
And
yeah, he's
I'm
he
gonna
yeah,
now,
yeah.
now, yeah, I just want to say, so we have about gone for about an hour, so we're gonna have to kind of finish the interview. But you practically answered. I want to finish with saying, like,
Did I
uh,
answer all your questions?
yeah, you did more than well, and I'd like we younger generations. I would like to finish to say that we really need people that can inspire us. And I think this story of Abelset, Amphotericin B, and how you chose to dedicate that to Leishmaniasis and the treatment of neglected global diseases, let's call it that. And it's been such an like wise, intelligent, informative conversation. I'm learning so much by it, and I'd like to really thank you for taking your busy time in your busy schedule to have this interview. So
Oh,
really,
it sounds my
thank you.
pleasure. It's great to support young folks like yourself who are doing podcasts. That's the way to be. I've only recently delved into the podcast world myself, and it's a wonderful world. So I know we're at the top of the hour, so I'll end there. If you have any other last questions for your audience, that would be great. And I hope I've addressed everything that you were looking
You
for.
you did that wonderfully, and I'll make sure to in the show notes put some links where people can check the university and the global health initiative
Yeah,
that you
there's
have there for
university
sure.
is global access policies. You can get the link there. The link to NGDI. You can link to some of the papers we're talking about that we've done recently. I've published about 70 papers just on amphotericin B.
That's amazing.
I mean,
That's
it's just
amazing.
ridiculous. I mean,
It's
I mean,
amazing.
as I
Mm.
said, and I'm actually going to MD Anderson Cancer Center at the end of March to give a talk, and I'm going to see my old professor, and I know he's going to give me a hard time. And it's like, you're still doing the same work 30 years later. What are you doing, man?
But
35
it's
years
me.
later. 35 years later. I
Yeah,
started
I
Dr. Lopez's
imagine.
lab in like 1988.
But
So what?
best of luck
37
with that,
years I've been doing
for sure.
the same drug, man.
Yeah, but it's for a good cause that we can be sure
Okay.
about. So thank you for the contributions you've made, and thank you, Dr. Kish, for this interview.
Okay.
Thank you so much for listening to this episode. I hope it brought you a new perspective, a moment of reflection, or even just a little bit of joy. You can find the video version of this episode on my YouTube channel, links in the show notes. And if you enjoyed the episode and would like to see more, please consider giving it a like or leaving a review on Spotify, Apple Podcast, or on my YouTube channel. Your support really helps others discover my content, and so, again, thank you for listening and for being part of this. I'll see you in the next episode of Medicine and Healthcare Around the World.